So the bacteria infected with virus that had the radioactive DNA were radioactive. So the DNA was genetic material that passed from virus to the bacteria. But when the bacteria infected with viruses that had radioactive proteins were not radioactive. So this indicates that the protein did not enter the bacteria from viruses. So DNA is therefore the genetic material that is passed from virus to bacteria. This indicates that the protein did not enter the bacteria from the viruses. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. Radioactive 35S sulfur S labeled protein capsule and radioactive P32 labeled DNA. The process infection, blending and centrifugation. There is a spinning word in the centrifuge. So this is the radioactive protein, and when it is infected, no radioactive 35S detected in cells. So radioactive 35S detected in supernant, but there is no radioactive in the supernant because the DNA has been passed inside the bacteria. That is, the radioactive 32P detected inside the cells because it contains the DNA that is passed. In here, this was the DNA that is being passed inside this. Histones are organized to form a unit of 8 molecules, that is a histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. These nucleosomes are beads on string on chromatin. In chromatin, not sorry. This one is the H1 histone. This one is the H1 histone, and so the 5' on phosphate side, and on sugar, that is OH 3'. So this is the phosphate sugar phosphate. This is the backbone, and the bases project inside like that. Bonds between the adenine and